Good afternoon, children.
Good afternoon, ma'am.
DNA fingerprinting. It's a quick method to compare the DNA sequences of any two individuals. 99.9% of the base sequences in all human beings are same. It's only the remaining 0.1% that makes all the differences. It makes each one unique. And while comparing the DNA sequences, you don't sequence the entire genome or the entire DNA sequence is not compared. Instead, only that 0.1% of it will be compared. Otherwise, it will be very difficult, expensive, and time consuming, because altogether we have 3 into 10 raised to 9 base pairs. 3 into 10 raised to 9 base pairs are present. So, in order to compare the entire genome, it will take so much time, and hence we concentrate only on that 0.1%. So instead of considering the entire genome, certain specific regions known as repetitive DNA sequences are used for this comparative study. So in human genome, there are certain DNA sequences which will get repeated. You call them as repetitive DNA sequences. So here in this particular figure, you can see this particular, I told you last period itself, GAAC. So like that, it is getting repeated 4 times. And here the same sequence is getting repeated 5 times. And here it is. Yes?
We can't see the screen.
Yes?
We can't see the screen.
You can't see?
No. We can't see the slideshow.
Wait. Now can you see? Now can you?
Yes, ma'am.
Yes. So it is repeated 8 times. First, it was in the first figure or in the first DNA sequence, it is getting repeated 4 times, then 5 times and 8 times. So these repetitive DNA sequences, only those are compared. So the repetitive DNA sequences, so in human genome, there are certain nucleotide sequences which will get repeated. And those sequences are compared for comparative study, for comparing the DNA of two individuals. And the number of times it gets repeated will be different for different individuals. Maybe a particular sequence, it is getting repeated 5 times in my sequence. Maybe it will be getting repeated 8 times in the other person, and 12 times in the third person, 50 times in the fourth person. So that is different. The number of times these sequences are getting repeated will be different for different individuals. And now you might be thinking, will it affect the coding or formation of proteins? But these repetitive sequences are non-coding, so it is not affecting the coding sequences. It is not, these are non-coding. DNA fingerprinting involves identifying differences in some specific regions. We don't have to compare the entire DNA, but those specific sequences where there are repetitive DNA, where a small stretch of DNA or that nitrogen base sequence will be repeated many times. And you can separate. So when you want to compare the DNA of two different individuals, you need to concentrate only on these repetitive DNA sequences. And for that, we can separate out these repetitive DNA sequences from the bulk genomic DNA. Bulk genomic DNA means the remaining, that 99.9%. So that you can separate by means of density gradient centrifugation. Density gradient centrifugation, already we learned about that in under the experiments. Cesium chloride density gradient centrifugation you had learned. So depending upon the density, they'll get separated, and once the centrifugation process is completed, two peaks will be formed, a major peak and a minor peak will be formed, and the major peak is formed by bulk DNA, and the minor peak will be formed by, so major peak by bulk DNA and minor peak will be formed by satellite DNA. Satellite DNA is same as the DNA having these repetitive sequences. So DNA with repetitive sequences, you can call it as satellite DNA. So once again, I'm repeating, when you need to compare the DNA of two different individuals, you don't have to compare the entire 3 into 10 raised to 9 base pairs, because majority 99.9% of the sequences will be same in all individuals, and just 0.1% will be different. That only we need to compare. And you can separate these repetitive DNA sequences or satellite DNA by means of density gradient centrifugation. And when it is carried out, it forms two peaks. The bulk DNA will be forming the major peak, and the smaller peak will be formed by satellite DNA. And satellite DNA, they are of two types. So satellite DNA means those DNA having, or the DNA sequences with repetitive sequences. It's of two types, microsatellites and mini satellites. So you can categorize, you can classify them into two types, micro and mini satellites. And this classification is based on the base composition, whether it is rich in adenine and thymine, or it is rich in cytosine and guanine, then based on the length and the number of repetitive units. So depending upon all these factors, you can classify satellite DNA into two types, microsatellites and mini satellites. Micro and mini satellites. These sequences are known coding. These repetitive, so even if it is repeated many times, it is not going to affect, because they do not code for any proteins, but they are forming a large portion of the human genome. So they are not coding for any proteins. And repetitive DNA sequences, they form the basis of DNA fingerprinting. We are able to carry out DNA fingerprinting with the help of these repetitive sequences, because they show great degree or high degree of polymorphism. I told you poly, several, morphism, forms, different forms, or genetic variability is known as polymorphism. And these repetitive sequences are different. The number of times these sequences are repeated will be different for different individuals. That is why it is said they exhibit polymorphism. So the number of times the sequences are getting repeated will be different in me and you. In each one, it will be different. So it is exhibiting polymorphism. Different people are having different times of repeat. So that is why we say they exhibit polymorphism. DNA fingerprinting was initially developed by Alec Jeffreys. He used satellite DNA as a basis for DNA fingerprinting. Children, we call DNA fingerprinting by this name because it is unique, just like our fingerprints. So just like our fingerprint, this is also unique. But the fingerprint, the normal fingerprint, can get altered by some plastic surgery, or when it is burned, or some acid is spilling over there. And different factors are there which can alter the fingerprint. But DNA fingerprinting cannot be altered, because the same copy will be present in each and every cell in our body. It's unique. It is the most reliable method of comparing the DNA of two different persons. Suppose in forensic sciences, just to identify whether the body belongs to a so and so person, or when there are paternity disputes, to prove that the child belongs to so and so person. For all that, we can use this DNA fingerprinting, because everyone will be having 50% of the DNA from father and 50% from the mother, so that there will be similarity between the DNAs of the child and their child and his or her parents. He used satellite DNA as a basis of fingerprinting, as it shows very high degree of polymorphism, because these repetitive sequences are showing great degree of polymorphism. Means it is different in different individuals. No two different individuals can have exactly same number of these repeats, so it will be different for different people. And he called it as variable number tandem repeats. Variable number tandem repeats. Tandem means arranged one in front of the other, one after the other. These repeats are arranged one after the other, and it is variable. It is different in different DNA, or in different people, these are different. So that is why you call it as variable number tandem repeats. See here in this picture, you can see how many times it is getting repeated. Twice repeated, thrice repeated, repeated 4 times, repeated 6 times. So it is these number of repeats is different in different people. Sequence will be the same. The sequence like GAG, CGG, that sequence will not have any change, but the number of times it is getting repeated will be different in different individuals. The technique involved southern blot hybridization. Now we will see how the DNA fingerprinting is carried out. What are the techniques involved? The technique is known as southern blot, or it involves southern blotting or southern blot hybridization. Here we are using a radioactively labeled VNTR as a probe. Probe means it's a fragment of DNA or RNA. It's a piece of DNA or RNA. Its length can vary, and you can label them radioactively. Why it should be labeled radioactively? Only then we can trace it out while carrying out autoradiography on an X-ray film. If it has to be visible, then it should be radioactively labeled. So the technique is southern blot hybridization. Let's see how is DNA fingerprinting carried out. To begin with, we need to isolate the DNA, blood sample, hair, saliva, anything. So from there, you can isolate from a single cell, the DNA can be isolated. In the picture, it is shown the blood sample. From there, the DNA is isolated. And once the DNA is isolated, then what is next step? It has to be broken down into small fragments. You call it as digestion. Digestion means breaking down DNA into small fragments. And last class, I told you what is the name of the enzyme which will help in breaking down DNA into fragments. Restriction endonuclease. It will help in breaking down DNA into small fragments. Then, now we need to separate these fragments. And for that, we carry out a method known as gel electrophoresis. In page number 198, that is under biotechnology, gel electrophoresis is given in detail. And in gel electrophoresis, what is done is we use, as the name suggests, gel electrophoresis, and the gel we are using is agarose gel, and it is extracted from seaweeds. It's a gel used for carrying out gel electrophoresis. And what is done is the DNA fragments. So the broken or the digested DNA fragments will be separated, or these fragments, you know that DNA are negatively charged. They'll be spread on this agarose gel. They'll be kept in agarose gel, and you know DNA fragments are negatively charged. Important to note, they are negatively charged particles, and an electric field will be applied. And upon applying an electric field, so fragments are placed in agarose gel and an electric field is applied. So being negatively charged, they will move towards the anode through this matrix. So they'll move through the agarose gel towards the anode. So being negatively charged. Once again, I will say these digested or broken down DNA fragments are spread on agarose gel. Agarose gel is a medium used for carrying out the process, and it is obtained from seaweeds. And then you apply an electric field, then you can find the DNA fragments moving towards the positive electrode, that is anode. Being negatively charged, they'll move towards the positive electrode, or they'll move towards the anode. And this agarose gel is having a sieving effect. They are having minute pores in it. And depending upon the size of the DNA fragments, they will move. So larger size, because they have to move through these pores in the agarose gel. So larger fragments, bigger fragments will move less. They cannot travel large distances, whereas those which are small, they will move longer distances. They can travel more, they can cover more distance. Hope you're understanding. So those DNA having so in this gel, those DNA having heavy fragments, so they will not be able to move further, whereas those which are lighter, they can move larger distances. So they will be moving according to its size. So smaller sized ones will be able to, the smaller sized fragments will be able to cover larger distances, whereas large sized one will not be able to travel farther. Okay. And in this way, based on the size, we separate out the DNA fragments. So the DNA fragments are broken down, then based on their size, they are separated by means of gel electrophoresis. They are broken down first using restriction endonucleases, then they are separated using gel electrophoresis. And then these DNA fragments, they'll be double-stranded. And these double-stranded fragments are made into single-stranded with the help of some alkaline chemicals. By using some alkaline chemicals, they are broken down, or their double-stranded will be separated and they become single-stranded. And then what is done? We carry out blotting. So when something is spilled, maybe ink has spilled somewhere, using a tissue paper, you will blot it out. Same way, using nitrocellulose membrane or nylon, using these synthetic membranes, you will blot the DNA fragments into it. You will press on this agarose gel, and from there the DNA fragments will get transferred into this nitrocellulose or nylon membrane. So from the agarose gel, they will be blotted. That's why you call it as southern blotting. Children, southern doesn't mean the direction over here, because this method was found out by E.M. Southern, the scientist. That's why it got the name southern blotting. So using nitrocellulose or nylon membrane, we are blotting these DNA fragments. Then after that, we are hybridizing them using a labeled VNTR probe. VNTR means variable number tandem repeats. And its probe means this probe will be having complementary sequences as that of the DNA fragments that we have separated out. So already we have so many DNA fragments. We have made it single-stranded. We have made these DNA strands single-stranded. Okay, and once this is completed, once blotting is completed, then we hybridize them using labeled means radioactively labeled. Why you need this radioactively labeled probe? Only then they will go and bind with its complementary DNA fragment, and we can see them. They can be visualized under autoradiography. You can see them on an X-ray film. So just like what you've seen in the figure, those line like you can see them on an X-ray film. So if it is not radioactively labeled, they cannot be visualized. So for that, we are hybridizing. Hybridizing means single-stranded one. The DNA is single-stranded. Now, what is done when it is hybridized with this probe? The probe with the complementary sequences will bind. This is our DNA fragment. This is the probe. So wherever the sequences are complementary, the probe will fuse with that, and that will result in hybridization. And later, this will be visualized using autoradiography. These hybridized DNA fragments will be detected with the help of autoradiography. Now, you might be thinking, why we need probe? Because only these probe you can label them radioactively. So once they are radioactively labeled, with the complement, they are having complementary sequences as that of the DNA fragments that we have. So these probe, when they are allowed to hybridize with these DNA fragments, they'll bind with all the DNA fragments which are having complementary sequences. And then the DNA as such, it becomes radioactive. So because of the radioactivity of this probe, the entire DNA fragment can be visualized under autoradiography. It can be seen on a X-ray film. VNTR belongs to a class of satellite known as mini satellite, variable number tandem repeats. I told repetitive sequences are generally known as satellites. Satellites are of two types, micro and mini. That also we learned. And VNTR is a type of mini satellite. Now, the differences between mini and micro satellite is that the repetitive unit, the size of so in micro and mini satellite, the repetitive unit, the sequence which is getting repeated will be between 2 to 9 base pairs. 2 to 9, it can be 2, 3, 4. So that sequence will get repeated. Whereas in mini satellites, the number of base pairs that are getting repeated can be between 10 to 100 base pair. So there the repetitive sequences are much more longer. These are short tandem repeats. Microsatellites are short tandem repeats. The sequences will be smaller. The repetitive sequences are smaller, ranging from 2 to 9. Whereas mini satellites, those are variable number tandem repeats. The sequences will be much more bigger, lengthier. So VNTR is a type of mini satellite, or it comes under mini satellite. A small DNA sequence is arranged tandemly, one after the other, in many copy numbers. And the copy numbers varies from chromosome to chromosome in an individual. The number of times it is getting repeated, the number of times these sequences are getting repeated will be different for different individual. And not only that, it will be different in different chromosomes of the same individual. That can be different from one individual to another. The number of times it is getting repeated will be different for different individuals. And not only that, within the same individual, different chromosomes can have different number of repeats. The size of VNTR is between 0.1 to 20 kilobase. And after hybridization with VNTR probe, the autoradiogram will give you many bands of different sizes. So we can visualize these repetitive sequences as bands. These bands give a characteristic pattern of an individual DNA. So each one will be having a particular pattern. It differs from individual to individual, except in the case of identical twins, monozygotic twins. Identical twins means from the same zygote, two individuals are being formed. For them, it can be identical, but in all the other individuals, this pattern will be different. The DNA taken from a single cell. You don't need so much DNA. Just one cell is enough to isolate the DNA. DNA fingerprinting is used in forensic science because every DNA, each and every cell is having the same DNA. The pattern will be the same in each and every tissue, whether it is blood, sperm, saliva, skin, bone. Any of these cells is enough to carry out the process. And the same degree of polymorphism, same time types of repeats, same number of repeats will be found in all the cells or tissues. So it is used in forensic science to identify the culprit. Then it is used for paternity testing when there is paternity dispute, as it can be inherited. I told polymorphism can be inherited from father to children, means both father and mother to children. It can be used for determining population and genetic diversities. So for determining population diversity, genetic diversity, and all this is useful. Children, see the first figure. There you can see the crime scene. From the crime scene, they got some blood sample, or let it be hair, or some sample, their skin samples they got. So from that, they have carried out DNA fingerprinting. They have carried out DNA fingerprinting, and this is the pattern of VNTR they got. The repetitive sequences. We are focusing only on the repetitive sequences. This is what we got. And 3 of them are suspected. 3 are suspected. And we collected DNA samples. So cells are taken. It can be blood sample or any cell sample. And DNA fingerprinting is carried out for suspect 1, suspect 2, and suspect 3. And then we are comparing how many times it is getting repeated in each one. So here you can see this is missing over here, and this one is matching with the DNA we got from the crime scene. If you compare, the number of repeats will be same. So maybe this is 12, 10. I'm just assuming some number. This is repeated 8, 9, 3. Simply I'm assuming some number. And if you compare here also it will be 12, 10, 8, 9, 3, 2. Here it can be some other number, 7, 3, 2, 4, 1. And here also 2, 3, 6, 9, 3, 1. So when you compare, you will find that this is best matching with suspect 2. So from that we can understand he is the culprit, and the sample that we have got from the crime scene belongs to suspect 2. That is how in forensic science they find out the culprit from the samples obtained. And then in the case of maternity dispute, and I told you children, 50% similarity with father's DNA and 50% similarity with mother's DNA. Now the child, mother, now we need to find out whether 2%, they are claiming that the child belong to those people. So father 1 and father 2, mother, then child. So DNA fingerprinting is carried out, and the child's DNA, we are comparing the DNA of the child with that of these 2 individuals who claim themselves to be the father of this particular child. So here also you can find, if you compare, this is first one, it's matching with father 2. Here also it is matching. And again this one, it is matching with the person 2. Here also matching is found, and here also. So it is more similar. It cannot be exactly identical to that of these the children, or the DNA of the children will not be exactly same as that of father and mother, but 50% similarity will be there. And when you compare, you will find that it is matching with the second person. So we can understand that father 2 is the father of this, the second person is the father of this particular child. And that is how we carry out DNA fingerprinting. Have you understood? Have you understood? Any doubts? Any doubts? 99%. 99.9% of the DNA sequences will be identical in you and me, but 0.1% will be different. And these 0.1% is the repetitive sequences. So some sequences, some DNA sequences will be getting repeated. Maybe it is repeated 50 times in me. In Gayatri, it is just 20 times. Then in some it will be, Nandana, it can be 2 times, 3 times. So it is different for each person. The number of times these sequences are getting repeated. Turn your videos on. Number of times these are getting repeated will be different for different individuals. That is unique for each person. So when you need to compare the DNA of two individuals, you don't have to focus on the entire DNA or the 3 into 10 raised to 9 base pairs. You have to focus only on these repetitive sequences. Remaining will be same. That is why in human beings, all the basic functions, basic metabolic reactions, all those are same because of this 99.9% similar genome. And we need to focus only on the repetitive sequences. And those repetitive sequences, by comparing the number of times these sequences are getting repeated, we'll be able to compare the DNA of two different persons. And you call it as variable number tandem repeats. Tandem means arranged one after the other. We call it as VNTR, or it is variable because it is not fixed for different individuals. It's different for different individual. It varies from one individual to the other. That is why you call it as variable number tandem repeats. Can I get some feedback from you? Have you understood? Say something. If you want some clarification, let me know. Okay, VNTR slide. Only that? Any other doubts? Malavika, have you understood?
Yes, ma'am.
Nandana, puzzled face. Have you understood?
Methodology.
Which one?
Methodology. I didn't understand.
Methodology. I'll repeat methodology once again. So VNTR, this one, Nia wanted to see. Nia, which slide you want to see? So methodology you wanted to see once again, no? Yes, this one, no? Nandana?
Yes, ma'am.
Nia, let me know which one you want to see. Where is the methodology? Yes, this one. First of all, we isolate the DNA. Okay, with the help of different enzymes, we will isolate, we'll separate the DNA. Just one cell is enough, and the DNA will be separated. Second step, this DNA will be broken into different fragments, small pieces. DNA is taken, broken into simple fragments. Okay? Yes? Here in the figure also you can find blood. From there you have taken the DNA sample using restriction endonuclease enzyme. We break it into different fragments. Then what we do, we carry out gel electrophoresis. When gel electrophoresis is carried out, the DNA fragments will get separated on the basis of their size. Larger fragments will not be able to move further, and smaller fragments will be able to cover large distance. So we can see them like a gradient. At the lower portion, the heavy or the bigger fragments, and smaller fragments at the tip. So gel electrophoresis will help in separating out DNA fragments on the basis of its size. Then what we do, you have to break it down, or you have to make these double-stranded DNA into single-stranded. So by applying some alkaline chemicals, the double-stranded DNA will be split into single-stranded. Okay, Nandana? Yes? Now what is done is using, because in agarose gel, it is lying. Now gel electrophoresis is conducted or carried out in agarose gel. So if you want to carry out further procedures in the gel, we cannot proceed. So this has to be transferred to some other solid medium. So we are blotting using is nitrocellulose membrane, synthetic membranes made of nitrocellulose or nylon. We blot this. Upon blotting, we press it on this, and these DNA fragments will get transferred into this synthetic membrane. Okay. Now, once that has been done, we have to hybridize them. Now, these DNA present on the nitrocellulose membrane are single-stranded. And if you need to visualize them, if that pattern you have to see, you have to visualize this pattern of DNA fragments, then we have to make it hybridized, join with a radioactively labeled probe, VNTR probe. Probe, as I told you, it's a small fragment or piece of DNA or RNA of variable length. which can be labeled radioactively. And why you want to hybridize it with this VNTR probe? Because only the probe can be radioactively labeled. With the help of that, we can visualize the DNA fragments. See, if you have to compare the DNA of two persons, we should be able to visualize it. Only then we can, on an X-ray film, this will be visualized. So only then we can compare. So for that, we make it single-stranded. Then we will be combining them with, we'll be mixing them with VNTR probe. So upon mixing it with VNTR probe, wherever the nitrogen bases are complementary in the DNA fragment, VNTR probe will go and bind. See, if this is the DNA fragment, these are the DNA fragment with nitrogen sequences, what we have got. So these VNTR, when VNTR is added, so wherever it is matching, it will go and bind. And whatever is not joining or binding, that can be separated out. And all these VNTR or all these hybridized DNA fragments, we can visualize with the help of autoradiography. Why we are able to visualize them? Because the probe is radioactively labeled. Because of the radioactivity, we will be able to see them when subjected to this autoradiography, or when autoradiography is carried out, we'll be able to visualize these DNA fragments. Nandana? Nandana? Okay? Your voice is breaking, Nandana. Any doubt?
Okay, last 3 points I don't understand.
Not clear? You message me. You message what is your doubt. Nia, which slide you wanted to see? Is this one or the next one? Is this one? Nia, please respond. Which slide you wanted to see? This one. VNTR is a type of mini satellite. Mini satellite and micro satellites are there. Mini satellite, here the length of the repetitive sequences will be longer, 10 to 100 base pairs. Whereas in micro satellite, it is only 2 to 6 base pairs will get repeated. Sorry, 2 to 9, either 2 or 6 or 7 up to 9. So those many sequences can get repeated. When it comes to variable number tandem repeats, 10 to 100 base sequences can get repeated. And the number of times it is getting repeated will be different for different individuals. Sequence is not getting different. In different individuals, it is not the sequence that differs, but the number of times it is getting repeated is becoming different. And then finally, once you get the autoradiogram, there we will see how many times it is getting repeated. And then accordingly, we will compare with the DNA of the other person. The copy number varies from chromosome to chromosome, even in an individual. Not only among different individuals it differs, but at the same time, even in same organism, you have maternal, and all chromosomes will be having 2 copies, maternal and paternal. So there also you can find differences. For example, chromosome number 7, you have maternal copy and paternal copy. So the number of times it is getting repeated will be different for both these chromosomes. So in a same individual also, there can be differences in the number of repeats in different chromosomes. Rijas, have you understood? Yes or no?
Yes.
Okay. Nandana? Nandana, is your doubt cleared? You have a message. Last 3 points from gel electrophoresis. Okay. Nandana, I will say, I saw your message. Nandana, just 1 minute. Listen here. So you want to know how the strands are getting separated. No? So there in these gel electrophoresis, the DNA strands are separated. You use some alkaline chemicals. It's not mentioned in the textbook. Some alkaline chemicals are used. Alkaline chemicals are used. Upon using these alkaline chemicals, the double-stranded DNA will get separated. By treating the DNA fragments with alkaline chemicals, they will get separated. Then what was the next doubt? Okay. Then what is done is you use a blotting paper. Nandana, where are you? Just turn your videos on. Nandana, are you there? See,
Yes,
once
ma'am.
in gel electrophoresis, the DNA fragments will get separated according to their size. Then we treat them with alkaline chemicals. So in presence of alkaline chemicals, a double-stranded DNA will get separated. Okay, then what is done? You use because further steps cannot be carried out in this gel medium. So you have to transfer it into a solid medium. So we use synthetic membranes made of nylon or nitrocellulose, and on the gel medium, we will press it. Okay, and we blot it. Blotting, you know, no? Like if something spills on the floor, using a tissue paper, you blot it off. Like ink spills on the floor, using a tissue paper, you're blotting it. So similarly, on this gel medium, nitrocellulose paper will be kept. And then these DNA fragments will get blotted. It will get absorbed on the nitrocellulose paper. Now, after that, what we do? We add it with, or we mix it with VNTR probe. And hybridization means now it is single-stranded. VNTR is also single-stranded. DNA fragments are single-stranded. VNTR probe is also single-stranded. So wherever these are complementary, they'll come and fuse. And VNTR is having radioactivity. So upon hybridizing, upon joining, so when they combine with VNTR probe, they'll become double-stranded. Okay? So the hybridization is taking place. So the entire DNA strand, that fragment, because of the VNTR probe, it will become radioactive because the probe is radioactively labeled. Then we subject it to autoradiography and we can visualize it and you can compare the DNA. Still, if you have doubts, I will explain in the next class along with the revision. We are starting with the same chapter for revision. So if you have any doubts, you note it down and let me know so that I can clear it in the next class. Okay? Yes or no?
Yes.
Nandana, at least some doubts I could clear? Okay. Okay. Anna is there?
Yes, ma'am.
Donna?
Yes, ma'am.
Donna is there?
Yes, ma'am.
Yes. Evelyn? Evelyn? Gayatri present. Lydia absent. Malavika is present. Meenakshi Bijoy? Not there. Mehak not there. Michael is there. Nandana present. Nevin is there. Nidhi? Nia? Naureen? Why don't you turn your videos on, children? Punya? Rijas? Sania? Sharon?
Yes, ma'am.
If you're not responding, I'll mark you absent. Steffi?
Yes, ma'am.
Anthony?
Yes, ma'am.
Vidur? Okay, thank you, children.
Present, ma'am.